Hello, my name is Louis Radigan, and today I will be giving you my pathogen presentation on influenza. Let's start with a basic introduction to influenza. Influenza is a virus that belongs to the Orthomyxoviridae family. It is enveloped, meaning that it has a lipid bilayer around it. It is segmented, which means that their genetic material is divided, and there are negative strands of RNA genomes, which means that it first must be transcribed into mRNA by RNA-dependent RNA polymerase, otherwise known as an RDRP, that has the virion in it to replicate, which actively spreads the virus throughout us. Out of the 4 genera for influenza, only 3 affect humans, with influenza A being the cause of multiple pandemics in the world, as well as seasonal flu epidemics. Influenza B being the cause of seasonal epidemics as well, and influenza C only causes mild illness when compared to the other 2. Influenza D only affects cattle and is not known to infect humans. Virulence factors include hemagglutinin, which is a glycoprotein that binds to sialic acid receptors on the surface of the skin, injecting itself into the host cell, and neuraminidase, which can degrade mucus, which allows it to penetrate the respiratory epithelium. It also helps with the release of newly formed viruses by cleaving the sialic acid made by the hemagglutinin, which helps with the transfer of the virus to other cells in the body. Influenza can be spherical or filamentous in shape, with spherical forming in 100 nanometers in diameter, while filamentous forms in excess of 300 nanometers in length. Internal proteins include the matrix protein M1, which holds the shape of the virus, matrix protein M2, which allows protons to enter the virus as it enters the host cell, and the RNA polymerase complex, which functions as the virus's RNA-dependent RNA polymerase to drive viral replication and transcription by making copies of the viral genome and viral mRNA. The way it works off of the 3 proteins that are listed on the slide here is that the PB2 protein snatches the cap from the host mRNA. The PA protein cuts off the capped fragment, and the PB1 protein uses the fragment as a starting point to synthesize a new viral mRNA, which allows the virus to replicate and make its own proteins. All right, let's go into the transmission of influenza. Influenza A is both a carrier type and zoonotic source, meaning that as long as it has a living host, whether it is a human or an animal, it can survive and thrive, spreading to other living hosts. Influenza B generally only infects humans, and influenza C only infects humans. Influenza cannot survive long in an environment outside of a host. The main form of transmission for influenzas A, B, and C is through respiratory droplets when an infected individual coughs, sneezes, or talks. It can also be spread through touching an infected surface and then touching your mouth, eyes, or nose. Alright, let's get into the flu overview, the symptoms, timeline, and impact. Influenza affects your respiratory tract, which includes your throat, nose, and lungs. Respiratory symptoms include a cough, sore throat, and a runny or stuffy nose. Systemic symptoms include a fever, chills, headache, malaise, which is a general feeling of illness and discomfort, and myalgia, which is muscle pain. Gastrointestinal symptoms include vomiting and diarrhea. The incubation period for influenza is in the range of 1 to 4 days. There are around 1 billion cases of influenza worldwide annually, with 3 to 5 million of those cases being severe. It causes anywhere from 290,000 to 650,000 respiratory deaths worldwide annually. People who are more susceptible to influenza are people ages 65 years and older, pregnant and postpartum women, children younger than 5 years old, and people with underlying medical conditions. 99% of deaths in children under 5 years old with influenza-related lower respiratory tract infections are in developing countries with a lack of resources to treat such illnesses. Now let's get into how to diagnose influenza. So the most common test for influenza is the Rapid Influenza Diagnostic Tests, otherwise known as RIDTs, which look for parts of the virus that stimulate an immune response, but they aren't as accurate as other flu tests. Another set of tests that is used is called the Rapid Molecular Assays, which detects genetic material of the flu virus and is more accurate than RIDTs. Tests in sterile environments such as hospitals and labs include a reverse transcription polymerase chain reaction, otherwise known as an RT-PCR, which converts RNA into complementary DNA, and then it goes through multiple cycles of PCR using DNA polymerase. Next up is a viral culture, which is where a virus is isolated and put in an environment to grow on its own, which helps us identify the virus, though this technique can take weeks and isn't recommended for rapid diagnosis. Finally, there's immunofluorescence assays, which use antibodies to detect viral antigens in the patient by lighting up under a fluorescent microscope where the virus is present. Let's get into influenza vaccines and treatment options. Influenza has a vaccination that people need to get annually because influenza goes through viral mutations quickly, making previous vaccinations less effective as time goes on. Antiviral medications such as oseltamivir, which is the generic brand name for Tamiflu, and zanamivir, which is the genetic brand name for Relenza, would help treat influenza and are the most effective if they are started within 48 hours of symptoms. Some support care options would be to stay hydrated, get rest, and to use fever reducers such as acetaminophen, which is the genetic brand name for Tylenol, or ibuprofen, which is the generic brand name for Motrin. Now let me talk about the advancement that I found in influenza treatment that I found really cool. Because there is a new study that found out that when an antiviral formulation in cacao plants called theobromine is paired with irinosine, which is a synthetic compound that interferes with the ability of cells to repair themselves, it targets proteins on the virus that serve as pathways for ions to travel in and out, which effectively kills the virus. It outperformed the current standard treatment of influenza, Tamiflu, in early testing, and a breakthrough like this could help form more effective antiviral strategies for future viral outbreaks, with the most prominent example of a viral outbreak that impacted the world very recently being COVID-19. All right, thank you very much for listening to my presentation.